Hi guys, so this is Maddie from your M315 class, and I'm going to walk you how to do each problem in the dilution worksheet. You're going to need to know this for your midterm exam as well as your final exam, because there will be a lot of dilution questions and how to back calculate and calculate your CFU per mil, stuff like that. Okay, so it's not too hard to it. Here's like a little cheat sheet on how to do these types of problems, or like the general formula you'll follow. So you'll see how all of these questions ask you to add a certain amount of sample, right, to an amount of diluent, right? So we're going to call this S, we're going to call this D, right? And we want to know what is the dilution that we just made. So a simple formula you can use is do S, so the amount of sample, over S plus D, right? So the amount of sample over the amount of sample plus the amount of diluent, right? So in this case, for problem 1, you would do 1 over 1 plus 9, right? So that's a 1 over 10 dilution, right? So a 1 to 10 dilution, right? Okay, so that follow. So let's try doing the second one. Just gonna erase all of this here. So for the second one, right, so if we're following our formula, which I'll write over here again, S over S plus diluent, right? We're doing 1 over 1 plus 99, right? So that's 1 to 100, right? So pretty simple. Okay, 2, 3. This one might be a little tricky because it's in decimal form, so we have 0.1 plus 0.99, right? So if we're following our formula here, we're doing 0.1 to 0.1 plus 0.99, right? So that is 0.1 to 1, right? But you can't just leave the dilution like that. You have to, I guess, how do I put it? Like, multiply this by 10 so that you have a normal, like, dilution ratio. And so what I mean by that is, it's weird to have a decimal in a ratio, right? Like, that's not a notation we usually use in math. And so to get rid of this, or to make it like normal, you actually have to get rid of this decimal place, right? So how do you do that? Well, we can actually shift this 0.1 and turn it into a 1 by multiplying this whole thing by 10, right? So 0.1 times 10 now becomes 1. 1 times 10 now becomes 10. So now your dilution is 1 to 10. And you can look at this on a calculator too. If you put in 0.1 over 1, right, you're going to get the same number as if you put 1 over 10, because it's the same ratio, right? All right, so getting rid of that. Hopefully you know how to do the rest of this point. I'm going to go over them very briefly since these are all the same type of problems. We'll quickly walk over to this, through this. So again, 0.5 plus 4.5, right? So that's 5. And then 0.5 over 5, right? So it's kind of similar to that 0.1 over 1 we saw earlier, right? So remember that this is a 1 to 10 ratio, right? Standard math here. Again, walking through this, so this is 0.3 plus 2.7 So that's a total of 3, right? So now we have 0.3 over 3, which is just like the last two problems, is just 1 over 10, right? Okay. Okay, again, number 6 is basically the same as number 4, so we have 5 plus 45, right, that's 50, and then so 5 over 50, right, that's a 1 to 10 ratio here, right? This one, hopefully you can just look at it and know, right? So 1 plus 19 here, that's 20, so this is a dilution of 1 to 20, right? 1 to 1, don't get tricked up here, right? This isn't actually a 1 to 1 ratio, right? We still have to add the numbers, so it's actually 1 plus 1, right? Which equals 2, and then so we have our 1 to 2 ratio here. So don't get messed up on these more simple problems here. For 9, hopefully you can also do this in your head. So we see 10 and 90, so the total is 100. And then we have 10 to 100, right? And that's, again, 1 to 10, right? And then finally, 1 to 49, our total is 50. So then we have 1 to 50. Okay, so I know I breezed through like the rest of those pretty fast, but I think here please like reach out to your AIs so that they can help you. Okay. All right, moving on. So these are basically the same problems, except in like diagram form, right? So more visual form. So in this case, right, here's our sample, right, and then here's our dilutant, right? So if we apply the same problem like last time, right, so remember the ratio is sample over sample plus dilutant, right? So in this case, it is 33 over 33 plus 99, right? And so it'll actually be, if I can draw that arrow, 33 over 100, 132, I believe, if I'm doing mental math right. And so this one might be hard to do the mental math for 33 over 132, but that's a, like 1 to 4 ratio here, right? Okay, so hopefully that made sense. Here's where it starts to get tricky, right? So here's our sample, right? But you'll notice now we have 2 samples coming from 2 different tubes, right? So don't get tripped up, right? We're going to take this one step at a time. We're going to calculate the dilution that's happening in each tube, right? So our first sample, right, S1, and this is our first dilutant, so dilution 1, right? And so we add those together, remember, just like last time. So the total volume for these two is going to be 5, right? And then we added 1 ml of our sample, so the dilution for this is 1 over 5, right? Okay, now we can calculate the dilution for this, right? So we're taking 1 ml again, and transferring it to 1 ml instead of 4 this time. So 1 plus 1, right? Our total volume for that dilution is 2, and we took 1 ml of our sample, right? So that's a half. So what do you do with these 2 dilutions now? You actually multiply them together to get your total dilution, right? So what I mean by that, I mean you do 1 over 5, so our first dilution, multiplied by 1 over 2, which is our second dilution, right? And then so you have 1 over 10 as your final dilution, okay? All right, so we're gonna do the same thing here, right? So for our first dilution series, right, we take 1 ml, we're putting into 9, so the total volume is 10, and we put 1 mil into that total volume, right? So it's 1 over 10 for the first one. This is the same exact thing, right? So it's another 1 over 10, right? And then here is where it gets a little tricky. How much you plate changes the dilution, right? So notice how this is in mils, right? So both of these numbers here are actually out of 1, right? So it's 0.1 mil out of 1, and then 0.5 mil out of 1, right? So if you remember back, this is actually a 1 to 10 dilution, right? And then this right here is actually a 1 to 2 dilution, right? So we're going to multiply the corresponding numbers together. So for dilution series 1, right, we had 1 in 10, 1 in 10, and another 1 in 10 dilution, right? So it's 1 in 10 cubed, so that's 1 over 1,000, right? And then for this one, it's actually 1 in 10 squared times 1/2, right? So it's actually going to be 200, right? Okay. Okay. So, sorry, the recording kind of... This one, so this is 1 over 100, right? This is 1 over 10. This is also 1 over 10, and then this is over 1, right? All right, so we just, multiply all of those, right? So 1 over 100 times 1 over 10, right, times 1 over, I can write 1 over 10, right? So these are the numbers that we calculated here. Okay, and then you add how much you added here, right? So when we put all this together, we'll have 2 over 10,000, right? Which we can reduce this down to 1 over 5,000. Or I believe the answer sheet also says 1 over 5 times 10 to the 3, which is the same number. So either way, either one of these would work. We put 1 mil into 99. Our total volume for this dilution is 100, and because we added 1 mil of our sample, it's 1 over 100 for dilution. This one, 1 into 9, right? So total volume is 10, dilution is 1 to 10. We added 1 mil of this, right? So this is 1 out of 1. So our total dilutions are 100 times, or 1 over 100 times 1 over 10 times 1, right? And so the final dilution is 1 over 1,000, right? And so what this problem actually wants you to do is calculate the original CFU per mil, right? So colony forming units per mil, right? And so we tell you here, hey, you have 50 colonies, right? And we calculated our dilution, right? So with these two numbers, you can actually combine them in this manner, right? You can do 50 colonies. I got 50 colonies in this dilution, right? So what you'll do is you'll actually take the inverse of this, right? So this is 1 over 10 to the 3rd. So it's 1 over 1,000, right? And you'll take the inverse of that, right? So the inverse of 1 over 10 to the 3rd is actually just 10 to the 3rd, right? So you'll apply that over here. So we'll do 50 times the inverse of our dilution, right? To get our dilution factor here. And so you'll have to reduce this number down, right? And to reduce it down, we just have to, you know, imagine the decimal places right here, right? Because this is actually 50.0, right? You can actually move this over by 1 by increasing the, what is it called? The exponent on the 10, right? And so if you have 50 times 10 to the third, it's actually the same as 5 times 10 to the fourth, right? Except this is like the most like reduced number, right? Okay, sorry, hopefully that made sense. Okay, so for this one, right, again, calculate your dilution, right? So we have 1 mil into 99, so 1 over 100, right? 1 mil into 9, so 1 over 10. 1 mil into 4, so 1 over 5. And then we took 1 mil of that and got 50 colonies, right? And so remember, calculate your dilution. So your dilution is 1 over 100 times 1 over 10 times 1 over 5, right? So that's 110 to the third times 1, 5, right? And so now we have, oops, sorry, pen back. So now we have 1 times 5, 1, 2, 3, right? So that's our total dilution, right? So we take the inverse of that to get our dilution factor, right? And so what we have to do is do 5,000 times, 50 times 5,000, right? And so, you know, you can do the math on that, and we'll have, you know, 250,000 colony forming units per mil, right? You can transfer this into scientific notation. Remember I was telling you about that decimal place? Well, you can actually count how many times you move it to get you your, to give you the exponent you need to add to the 10 for your scientific notation. And so what I mean by that is you can count literally 1, 2, 3, 4, 5, right? So this number in scientific notation is 2.5 times 10 to the fifth, right? Because we move that over 1, 2, 3, 4, 5, right? So that's the answer here. Hopefully that made some sense for you. Okay. Again, similar thing here. Actually for 17, so the answer is supposed to be, I guess there was some confusion as to whether they, you need to average these two numbers here. And I can show you like the answers to both. Ideally, what will happen is that for one number, we're going to get 1.45 times 10 to the 7th, and for the other calculation, we'll get 1.35 times 10 to the 7th. And so if you average those, you get 1.4 times 10 to the 7th. So normally in actual like labs, we don't do averages like that. Typically, you just, you know, pick one plate and count the CFU per mil from that one plate. But in this case, you know, both 1.45 times 10 to the 7th and 1.35 times 10 to the 7th are very close to 1.4 times 10 to the 7th, right? Okay, so let's actually get to like the math on that. So we have 1 into 99, right? So that's 1 to 100 dilution. Again, another 1 to 99, so that's another 1 to 100 dilution. And remember how we did these, right? So this is over 1 over 1. So this is another 1 to 10 dilution, whereas this is, 2 to 10, so 1 to 5, right? Okay. So we're given the colonies for each of these plates, right? So for the, where should I write this? The 145 colony, right? For that number, you're going to do 1 for over 100 times 1 over 100 times 1 over 10, right? And so your dilution is 1 over, let me see, 1, 2, 3, 4, 5, 10 to the 5th, right? So this is your dilution. Your dilution factor is the inverse, right? So your dilution factor is 10 to the 5th. And so for the 145 plate, you would do 145 times 10 to the 5th, right? And then you reduce that down. So now it's 1.45 times 10 to the 7th, right? So that's one of the numbers. For our 270 colony plate, right, it was almost identical, so we still did the 1 over 100 times 1 over 100, except this time we didn't do a 1 over 10 for that plate, we did a 1 over 5, right? And so what happens is you have a 1 over, like 50,000 dilution, right? Remember, we take the inverse of that and multiply our colonies by it to get our CFU per mil. So it's 270, right, times 50,000, right? And so that will give us the 1.35 times 10 to the 7th. So again, this problem is kind of weird because ideally you would have rounded and saw that both of them went to 1.4 times 10 to the 7th. Like this, technically, if you round that, it's 150. But like on the actual exam, you will not be averaging like colonies. They should both give you like the same CFU per mil, ideally, right? So ideally it would have said 1.4 times 10 to the 7th. So if this problem confused you, it's not because you didn't know what you were doing. It's because technically we put the wrong number of colonies here. It probably should have been more like 140 colonies to get you the this answer, right? So don't freak out. Okay, so moving on to 18. So again, calculate your dilutions, right? So 1 into 99 is 100. 1 into 9 is 10. 1 over 10, and then another 1 into 9 is 1 over 10. Remember, these are out of 1, right? So this is just 1, and this is actually a 1 to 10, right? So here's the kind of trick question part. You see how we gave you 3 different plates, right? So one has 600 colonies, one has 10, one has 80, right? And if you try to do the math on all 3 of these, they're going to give you a different CFU per mil. Now, what do you do, right? What do you do in this case? Well, remember in lab, we only count plates that have between 30 and 300 colonies, right? We only count plates that are in between that range. If it's too high, it's called too numerous to count, right? And we don't use it to calculate our CFU per mil. And if it's too low, we say it's too few to count, right? And we also don't use it. So because This plate has 600 colonies. We're not going to use it in our calculation. And because this plate has only 10 colonies, we're not going to use that plate. 80 colonies is in between the range we set earlier. So we're going to use this number to actually calculate the CFU per mil, right? So in this case, it would be, let's see, where's the best place to write it? I'm just going to make a little box here. So we do, you know, our 1 over 100 here, right, times our 1 over 10 times 1 over 10, right, and then the amount we plated was just 1 over 1, so that's just 1, right? So our dilution is 1 over 1, 2, 3, 4, right, or 1 over 10 to the 4th. And remember, our dilution factor is the inverse of that, right? So our dilution factor is going to be 10 to the 4th, right? And so you can now do 80 times 10 to the 4th. That will give us our CFU per mil. Remember, we have to reduce this down to 8 times 10 to the 5th, right? So that's the answer for this problem. So our original culture had 8 times 10 to the 5th colonies, or CFU per mil. Okay, so this one, again, just add these two numbers. So you're adding the sample and dilutant, right? So we have 21.6 of the dilutant, and then we have 2.4 mils of our sample. And so that just equals out to 24, right? And then so our dilution is our sample over the total volume, so 2.4 over 24, and so that's just a 1 to 10 dilution, right? So D is the right answer here. Okay, and then for this one, a person wants to make a 1 to 1,000 dilution, right? This also can be written like this. And you basically have to figure out which of these dilutions would not give you a 1 to 1,000 dilution, right? So easy, right? So just add the number of sample to the amount of dilutant, right? So 10 plus 9,990 mils of water will give you 10 to 1,000, right? So so this, sorry, 10,000. So this would give you 1 to 1,000, right? Because 10 plus 9,990 would get you to 10,000 mils total, right? So this is not the correct answer. Well, we'll give you the 1 to 1,000 dilution. We're looking for the ones that won't give you it, right? So just to be clear about that. So again, add this, right? So we have, it's basically like the same problem above, right? So 0.01 plus 9.99, right? So that's also going to be a 1 to 1,000, right? Okay, so this one is going to be 0.05 to 49.95, right? So the total volume is going to be 50, and so 0.05 to 50 is also a 1 to 1,000 dilution, right? Okay, and then, so this one, right, place 0.1 mil of an undiluted sample into 9.9 mil, right? So what you'll notice for D is that this, the total volume, right, is 10. We use 0.1, so 0.1 to 10 is only a 1 to 100 dilution. It's not 1 to 1,000. So D is the correct answer for 20 because the rest of these will give you a 1 to 1,000 dilution except for D. D will only give you a 1 to 100 dilution. Okay? All right. So 21, so a tube containing 5 mils of broth, you add 0.1 mil of solution that has 50,000 units per mil. We want to know how much penicillin, what's the concentration of penicillin in the broth now, right? And so hopefully you remember from like your chemistry classes, but M1, V1, or I guess even physics does this too, N2, V2, right? And so our tube has 5 mils of broth, right? That's our volume. We have our known concentration, right, which is like 50,000 units per 1 mil, right? And we know how much of that stock solution we added, right, which is 0.1 mil, right? And so we want to know the concentration in the 5 mils. So this is our unknown. This is our x, right? And basically you do simple algebra, solve for x, right? X equals 1,000, right? Pretty, oh, sorry, wrote then. My bad. It's late at night. So that means C would be your answer. Right, so I believe you guys are smart enough to do simple algebra. I don't think I need to walk through that, but when you're asked a question like 21, just remember this M1V1 equals M2V2, and it should be pretty simple to solve, okay? All right, so for this one, we have 1 gram of sample soil added to 9 mils, right? So that is already a 1 to 10 dilution. We can write that down somewhere. 1 mil of that mixture is placed into 99 mils, right? So that's another 1 in 100 dilution, right? And then from this, 1 mil is plated, so that's just 1/1, so we don't need to care about that. And then we get 200 colonies from that, right? And so it wants us to back calculate the original number of colonies in the soil, right? And so remember, we need to calculate our dilution to get our dilution factor, right? So we have, this is our dilution, right? So that is 1/10 third right there. And then the dilution factor is the inverse, right? So 10 to the third. And so we multiply our dilution factor by the number of colonies we see on the plate, right? And then we would reduce this down so that it is 2 times 10 to the 5th, right? So C is the right answer here. And again, I think you guys are smart enough to know this, but just in case you haven't learned this, trick yet, how you can easily calculate, go from this to that is we start with 10 to the third here, right? And then we want to move the decimal place here 1, 2, right? And so you add to the exponent here, right? And so that's how we get 2 times 10 to the 5th, right? Hopefully that made sense. Okay. Getting close to being done. Okay, so this, pretty simple, right? So this is 1/100, right? 1/10. And then this is, remember that this is out of 1, so this is actually 1/10, right? So we want to calculate the bacteria per mil, or colonies per mil. This should actually say, because 1 colony does not equal 1 bacterium. But okay, so let's see. So remember, we multiply our dilutions, right? And so we have 1, 2, 3, 4 zeros. So this is 1, times 10 to the fourth, right? Our dilution factor is the inverse of that, so just the inverse of that, so it's 10 to the fourth. They tell us we got 45 colonies, so remember we do 45 times 10 to the fourth. We have to reduce it down to the single decimal place here, so it's actually 4.5 times 10 to the fifth, right? And so we look, that one is D, right? Okay. Technically we need to write like CFU per mil to be accurate. Okay, cool. So there are 2 times 10 to the 6 bacteria in 1 mil of suspension. You dilute the bacteria by a factor of 10 to the fourth, right? How many bacteria would there be in 0.2 mils of this dilution, right? So this is maybe a little more complicated than the problems we've seen before, but let's just write everything out, right? So 2 times 10 to the 6 is what we start with, right? We just write the units so that we're clear. So we have 2 times 10 to the 6 CFU per mil, right? You dilute this by 10 to the fourth, right? So do the math, right? You minus the exponents in this case, right? So 6 minus 4 equals 2. So now you're at 2 times 10 to the 2, right? CFU per mil. And so, let's see, you plate only 0.2 of this, right? Remember, this is out of 100, oops. So, sorry, this isn't out of 100, this is out of 1, right? So 0.2 over 1, so that's actually 2 over 10, which is 1 over 5, right? And so what we would do is this, do this number times, 1 over 5, and you should get 40, right? Okay. Pretty simple. So do it in a different way, right? And then remember 0.2 over 1, that's equal to 2 over 10, right? So 2 over 10, right? So you actually take away this, so now this is just 10, right? And then 2 is still, in the numerator, right? So 2 times 2 times 10 is 40. So hopefully one of those two ways made sense to you. Okay, so for 25, we have our 1 gram sample of soil to 99, so that's 1 in 100 dilution. They do the same thing again, so 1 over 100. From this dilution, you plate 0.1, so remember this is over 1, right? So that's 1 in 10, right? And then from this dilution, you plate that, and then you get 200 colonies, right? So remember, we need to calculate our dilution. So it is 1, 2, 3, 4, 5. So 1 times 10 to the fifth, right? Our dilution factor is the inverse, so 10 to the fifth. We do 200 times 10 to the fifth. We have to reduce this down, right? So now it's 2, and then 2 times 10 to the 7, right? Okay. Alright, so I think this is, we're getting close to being done. Okay, so you are given 2 times 10 to the 9th bacteria per mil. You want to only get 100 colonies, right? So remember I talked to you about like the dilution factor and how you can actually use the dilution factor to figure out, I guess this like, better just show you like this. So we have 2 times 10 to the 9th, right? To get 100 colonies, right, we need to be in the 10 to the second range, right? Because that's 100, right? And so we need to dilute this down and get this exponent to 2, right? So if we dilute this by 10 to the 7th, right? Remember, when you do this, you actually minus the exponents, right? So you get 2 here. Now you would be at 2 times 10 to the second, right? But you're still too high here, right? Because you're at 200 colonies when this happens. So if you plate half of this, right, 0.5 mils instead of 1 mil, right, or a dilution factor that gets you to half, you'll get only 1 times 10 to the second, which gets you to 100, right? So we need to find out which one of these fulfills like all of these requirements here that we just figured out, right? So remember I said that diluting by 10 to the 7th is immediately going to help us get within range, right? So we can cross these other guys out because we know they're wrong. So it leaves us between A and D here, right? And now we need to figure out, how do we get to this final dilution factor of 1/2, right? So we can look at A. So this is 5 into 10, right? So our total volume is 15. So 15, 5 into 15 obviously isn't 1/2, right? It's 1/3. So that's A is not right. And we can just verify that D is right because 5 plus 5, so we have 5 into 10, and that's 1 into 2, which is the dilution factor we were looking for, right? So D is correct. Okay. Okay, by the way, you can skip 27. We didn't teach you how to do the conversion factor for those like counting chamber stuff, so you don't have to know how to do 27. We're not going to ask you how to do that. So moving on to 28. So you are given food that has this range, right? So 5 times 10 to the 5th and 1 times 10 to the 7th, right? Okay, so again, kind of like the last time, we want to plate 1 mil of this. We want to get our final, colonies on our plate, our final dilution, to give us between 30 and 300 colonies on a plate. So it's very similar to 26, right? And so we have 5 times 10 to the 5th here, right? So if we want to be within this 30 to 300 range, we need to be between 10 to the 1st and 10 to the 2nd, right? For our final kind of like CFU per mil, right? And so to get into that range, we need to dilute it by 10 to the 4th, right? Remember that we minus the exponents when that happens, so that we would end up with 5 times 10 to the 1st, which is just 50, right? And so 50 colonies is within our range here, right? It's within that range. So we know that 10 to the 4th is going to be in this answer, right? So immediately we know it's going to be like one of these guys here, right? So we knocked out A and E. Okay, but we still have to figure out the dilution factor for this, right? And so if we tried to do 10 to the 4th again, remember 4 minus, or 7, 7 minus 4 is 1 times 10 to the 3rd, right? And that's 1,000. That's too high for our range, right? So let's try diluting down by 10 to the 5th. So 7 minus 2, right? So now we're at 1 times 10 to the 2nd. We're at 100 colonies, which now fits within our range. So these are the 2 dilution factors we needed. So the answer is D. so I believe this is the last problem. Yeah, okay, perfect. So, very simple, right? So, just calculate your dilutions. This is 1 in 100, this is 1 in 10, this one's 1 in 10, this one is just 1 in 1, 1 in 1 here, and then remember 0.1 over 1 is just 1 over 10, right? And so remember, just like with, let me check, just like with problem 18, you need to decide which of these numbers to use, right? So remember, the ideal range is 30 to 300 colonies. So 940 colonies is too much, don't use that number. 10 colonies is too few. 150 is just right, right? Okay, and so we already calculated our dilutions here. So let's just do the math. So our dilution is 1 over 100 times 1 over 10 times 1 over 10. And you plated 1 mil, so that's just 1 over 1, right? And so it counts as zeros, 1, 2, 3, 4, right? So our dilution was 1 over 10 to the 4th, right? So that means our dilution factor, which is the inverse, is 10 to the 4th, right? And now we multiply our number of colonies times the dilution factor to the 4th, and we have to reduce it down, right? So it's 1.5 times 10 to the 6th, right? And let's see, that is B here. Okay. Cool, that was the last problem. Hopefully that helped a little bit. Hopefully that didn't just confuse you more. All this stuff is relatively easy once you know how to do it. I think you guys can do basic dilutions. It's just problems like these, please remember to choose the right plate, or else you're going to get really wonky numbers. You're going to get the wrong answer, right? So really, I feel like you guys just might get stuck on that. So just remember this, that we want to only count plates, only count colonies, or use the colony number from plates that have between 30 to 300 colonies, okay? All right, awesome.